Morning, students. Good morning, everyone. So today we will see the important aspects of body fluids and circulation. So this lesson is quite easy lesson, but there are a few topics which you need to stay there and you should be focusing, like cardiac cycle, events in cardiac cycle, and the times. At the same time, valves, layers, and their functions, right? Uh, so especially heart sounds. What causes the heart sounds, that is also important. So especially you need to focus on the cardiac cycle more. So the cardiac events which occur, and during that cardiac event, what are the different aspects that occur in the heart that you need to focus. And of course the diseases related to circulation. So these are the main aspects. So we need not go in depth of other topics. For example, double circulation is there. It's a very surface topic, where you can see that how the systemic circulation, pulmonary circulation is being simultaneously going on. So those are all you can self-explanatory. You can just read by yourself. So, but where you need to stay focused is nothing but the cardiac cycle, right? So let's at the time when we talk about this, what are the other things that we can really look into this lesson? Blood groups. When we talk about blood groups, we already in principles of inheritance and variation, codominance, you would have multiple alleles. You would have studied the blood groups also. So those blood groups on just check, right? So who is a donor? Who can be donor? Who can't be donor? What are the different combinations we can get it, right? So that you need to focus. So because they may be asking some questions related to that as well. And when it comes to compositions, blood composition, plasma composition, what are the other aspects that present in the plasma? At the same time, their functions can be asked as correct or incorrect statements. Okay. So we have, when we talk about the formed elements, we can talk about, we'll be talking about, erythrocytes, thrombocytes, we have white blood cells, right? So within the white blood cells or leukocytes, you have different types of leukocytes, which actually involves in immunity, providing immunity, right? So how you have to do is take each of the cell, like lymphocyte. What is its functions? What are its functions? What is the shape of it? Monocyte, shape and function. Eosinophil, shape and function. Whether they are nucleated or enucleated, also you need to focus, right? Especially their formation, where exactly they form. So that is one of the aspects you need to focus, because the cells, immune cells, from what is the location of them would be the important aspect that you need to once quickly go through, right? So in RH blood grouping, right? So pregnancy, when especially the pregnant woman, how can she pass? Like how this RH incompatibility can lead to the death of the fetus, right? Erythroblastosis, right? Erythroblastosis fetalis. What conditions does this erythroblastosis fetalis occurs? Right, that you need to focus. There are n number of you can have different combinations also. If mother is Rh positive, if the child is Rh negative, if mother is Rh negative and child is Rh positive, and during first pregnancy and during next pregnancy, and how can we introduce this what is a how can we combat this situation, right? So that you need to focus in this. And today's session would be not that much long, because this is quite a very easy chapter, and the things that you need to focus on, few things, okay, that I will let you know. Especially clotting of blood, if you see in the NEET book, you will be having the clotting factors. So clotting factors and their functions, you need to focus. For example, clotting factor 1 is there. So clotting factor 1, that is fibrinogen, and what are its characteristics, right? And if you see that it is a glycoprotein and it is synthesized in liver, right? So what are the different functions of this fibrinogen? For example, factor 2, prothrombin. So you would have heard about prothrombin, thrombin, and this formation of this clotting, etc. So you need to focus on what are these factors, what are these names and their functions. Especially the clotting, whatever the clotting, the sequence of events, cascade of events that takes place during the blood clotting, that is extrinsic and intrinsic pathway, which leads to the clotting of the blood, right? So that you need to focus, because they may give as a flow chart, and they may remove any one of the factor, and they may ask that what factors does involved in this pathway, right? If there is a damaged tissue, right, few plasma factors would be released, right, and that leads to what? So this flow chart you need to actually follow. So at the same time, prothrombin converts into thrombin, by which which is necessary over there, and what are the inactive prothrombin does, and what thrombin does is thrombin again it could be eventually converted to fibrinogen, and then fibrin, and then finally blood clot. So in this way, you need to approach these flow charts, right? So especially the blood clot is concerned, these blood clotting factors are very, very important, right? So then when we talk about there is a chance that you may be asking, you may get questions like column A and column B. You'll be having 2-chambered heart, 3-chambered, 4-chambered heart, and their examples. So across these vertebrates, how these hearts, right, is varying. So that you need to focus. In NCERT also they gave straight away they gave few examples. Double chambered hearts, at the same time 3-chambered heart and 4-chambered heart, and exceptional cases in reptilia also you need to focus. So those examples are important, and especially where do you see single type of circulation, where do you see double circulation and incomplete double circulation also you need to focus. Okay, then only you will understand that the evolution of heart, fishes to amphibians to reptiles to birds and mammals, there is an evolution of heart we can see. So that you need to stay focused. So there is incomplete and complete double circulation also. So there is a mixing of blood also would take place. So I'm again reminding you guys, I'm not explaining anything here, anything in depth. I'm just giving an important areas that you need to cover. Just quickly, I'm making you rewind what already you have learned for 2 years, right? So these are the aspects that you shouldn't be ignoring. So there is an incomplete double circulation, complete double circulation, and you have single circulations also. So where does this single circulation occurs? Where does incomplete double circulation occurs? Where do you see complete double circulation? Is the these are the important aspects that you need to cover. Right? And if you see this heart, in the heart, the muscles are concerned, you have this tissues are concerned, you have cardiac tissue, at the same time you have nodal tissue. So this nodal tissue and their parts, for example, if you consider this is a heart, right? So the left this right atrium, you will find SAN node. So from the SAN node, which is a pacemaker, you will be getting impulses, and it will be traveling to the AVN, and then it will go into the bundle of His junction, and then it will go into Purkinje fibers. So this pathway of the nerve impulses, these the electrical impulses that travel right down or right from the SAN node pacemaker. Extremely important. So this Nordahl tissue, you should be practice with a rough diagram, just like it's a branching thing actually. So you need you just focus on that. That is Nordahl tissue. So Nordahl tissue, don't ignore, right? Nordahl tissue and cardiac tissue. So in cardiac tissue, what are the different types of we have layers. We have pericardium, myocardium, endocardium, and their positions and their locations and their functions also you need to focus. Okay. And coming to the heart, structure of heart, right? In structure of heart, what you need to focus is that the valves, the names of the valves and their location and their functions. Okay. So what are semilunar valves? What are tricuspid, bicuspid valves? Right? What are their functions? So those you need to just brush up those things. So that's what when I talk about Nordahl tissue, that's a conducting system of the heart, where exactly this impulses starts, SAN node and then AV node and then AVN node. We have a bundle of His and then Purkinje fibers. So entire this Nordahl tissue is very much important. And coming to the hormones, AVN hormones will be there, right? This AVN hormone, you would have remembered that in chemical coordination we studied this AVN hormone which secretes in the heart, right? So that function you need to just brush up. This hormone, these are all where you need to have which are conceptual at the same time memory based, right? If you don't know this, if you don't remember this this words or their functions, you can't really tackle few questions. So brush up that. So we have that hormone, right? So that we have this ANF, that is, you just try to focus on this, try to recollect this ANF. It's not ANV, it is ANF tissue, ANF factor, atrial natriuretic factor, ANF and their functions. Try to focus. Okay. And then coming to the side, cardiac cycle. So these events and the time taken and step by step process. That means one phase to other phase. This flow chart is important. It means atria is there, ventricle is there. So atrial systole and diastole. You should be very much careful with this systole, which would be a contraction, and diastole, which would be a relaxation. So this contractions and relaxations of atria. So atria is there, right? So you consider this the upper chambers of atria, those are all systolic, like contractions and ventricular contractions. So this auricular or atrial contraction, how many seconds it will take place? At the same time, how many seconds it will be relaxed? And when this contraction is taken, what is the position of ventricle? So those things you need to focus. So approximately the time of a cardiac cycle would be 0.8 seconds, approximately. So that's what 72 times per minute, the heart rate we can calculate. So regarding the heart rate, you may get a few numericals, very simple numericals. You may expect whenever there is a rate that you have to really careful about those type of questions. Okay. So events that occurs during a cardiac cycle would be at most important. So ventricular systole, what are the events? right? And in atrial systole, what happens, right? Blood would be filled, or it would be pumped out. So those things you need to focus. And coming to heart sounds, the lab-dub, what is the first heart sound and the second heart sound? So first heart sound is it would produce by closure of the, just like imagine that you will be closing the your room door, then you will be getting a certain sound, right? So there would be flaps like structures, so they will be like this, and they will be closing and opening, right? So during the closure of this bicuspid and tricuspid valves, you will be first hearing the first sound, right? And this is low pitched, less loud, right? And longer than the second heart sound. But the second heart sound is produced by the closure of aortic and pulmonary semilunar valves, which is louder, right? But the time is less. No, it lasts for only 0.1 second. So these are the little differences you need to focus on when it comes to heart sounds, right? So when we talk about the regulation of heart's functions, so how the hormones can actually regulate the heartbeat, right? Those things you need to once brush up. Those are all facts actually. So when you talk about the cardiac cycle, the calculation of cardiac output, so you may expect numerical problems from cardiac output, right? So how do you really calculate the cardiac output? So it is a normal rate of an adult per minute into amount of blood pumped by the heart per minute, right? So that's how we actually calculate the cardiac output, and this cardiac output may vary in different circumstances, right? So these are the aspects you need to cover when we talk about the cardiac cycle. So within this, cardiac output is important, systole, diastole, and their timings and their functions. What exactly happens during the atrial systole and ventricular systole, right? And then coming to the EKG or EEG, electrocardiogram, right? Who got this Nobel Prize that you need to once look into it. So here I will be typing that name. Okay. So Intoven actually got the Nobel Prize, right? So Intoven got the Nobel Prize, considered to be a father of cardiography. Okay. So just you would have first time heard. So Intoven, who is a father of electrocardiography. So I try to remember this name. And most importantly, in this ECG, the PQRST, especially this peaks and this spikes, where exactly this what exactly each segment represents, right? Like P wave, Q wave, right? R and S wave, T wave, what they actually signify, right? So in this is where you will be talking about depolarization, repolarization. All right. So here each peak that you need to study. Why? Because sometimes they may give call me questions or correct or incorrect statement. What is correct statement regarding P wave, right? QRS wave. You have right. They may be asking in this QRS wave segment, and when what starts at the end of the T wave, what starts actually. So these are the things you need to once try to focus in this, right? So at the same time, in NEET book they will be given even in abnormalities of ECG, right? What happens if P wave is abnormal, Q wave is abnormal, so just have a look at that. But you need to focus on QRS, what is what happens there, whether there is a ventricular depolarization or repolarization. So repolarization that you need to once you have to try to draw that and try to assign the functions to the each wave. So that would be more easy to recollect the stuff. Okay, and parts of the artery, different cross section of the artery also once check that. So what else we can really talk about in this is next comes this the final aspect can be of different heart conditions, right? So different disease conditions or disorders of circulatory system, right? So we have different disorders. So each disorder name and its symptoms, right? So those things you need to focus. Few things would look very similar, so you should be very much careful, right? Atherosclerosis is there, coronary artery disease, CAD is there, right? Heart attack, heart failure, right? They look similar, but they are little different when it comes to the condition. Heart attack is a myocardial infraction, right, where it is a death of a part of the heart muscle, right, which follows a cessation of blood supply. So what about heart failure? It is a condition when heart does not pump blood efficiently enough to meet the need of the body. What actually need of the body is oxygen needs to be given and the CO2 has to be taken up. So when the heart is not efficiently pumping, that is heart failure. Heart attack is something myocardial infraction. So we have already talked about the layers, tissue layers of the heart, cardiac tissue. So that's what you need to focus. So these are the different aspects that you can really brush up on. So more than we covered entire lesson here, but the thing is, again, you need to go through the cardiac cycle, events that occur, depolarization or repolarization, whether it is in ventricular or auricular repolarization or depolarization, timings. Again, I'm posting previous year's questions. You can use them as a practice questions also, so that it will be easy to get an idea of what kind of questions you may get in this lesson. So when physiology is concerned, human physiology, almost 6% of questions may come from each lesson. So don't ignore this lesson and quickly complete these factual parts and then functional parts. Factual part like ANF and their functions, location of SAM node, right, and location of Purkinje fibers, right. What are their functions? These are all memory based questions, right. So more than this, nothing to be dealt in this, right. So if you have any doubts and if you need anything, any topic of explanation, please ask me. Otherwise, we can end this session, because these are the important aspects. The human physiology lessons, mostly, this neural coordination, chemical coordination will be lengthy with more concepts. So there we will stay there and we will I'll try to explain a few concepts also. But in this, you can self-explanatory. You can read by yourself, and this lesson is not that much tough as well. But if you feel any topic which is tough, you can ask me here, so that right now I will discuss about it. Otherwise, we can end the session. Is that okay? So any doubts or not, you let me know. Okay then. Yeah, prepare well for tomorrow's test, CT9, I guess. Yeah. So all the best. Do well. Take care. Stay home. Stay safe.